Hi, I'm Sam Hoare from Pharmacanics. In this video, we're going to talk about GPCR signaling assays, specifically the pharmacological data we get from these assays and how we interpret the results. Here in this graph, we have cyclic AMP generation being stimulated by an agonist via a GPCR. These data can be explained by a fundamental theory of GPCR pharmacology, the operational model of agonism. The goal of this video is to explain this theory in simple terms to enable us to better understand our GPCR pharmacology data. In a follow-up video you can find on the Pharmacanics YouTube channel, we dig into the operational model in more detail to explore and understand how the model parameters affect the data we obtain. Let's expand the graph data here. Here we are varying the expression level of the GPCR. We are testing the same agonist on 4 cell lines with varying levels of receptor expression from high to very low. This shows us that the response to the agonist changes dramatically with the GPCR expression level. This is a typical observation in GPCR pharmacology. This behavior is beautifully explained by the operational model, a pharmacology theory which was first published in 1983 by Sir James Black and Paul Leff, with input from Terry Kenakin and others. This model emerged from the field of receptor theory as the culmination of many decades of receptor theory development, which you can read about here. This model was derived before we understood how GPCRs function, before the isolation of G proteins and the cloning of receptors. This leaves us with a problem. The operational model is often explained in the mathematics and language of traditional receptor theory developed over 50 years ago. This theory, while highly successful for drug discovery and fascinating for people like me, can be quite impenetrable to the modern drug discovery scientist, as you can see from this diagram. As a result, the drug discovery scientist often has little understanding of how GPCR pharmacology really works. Fortunately, the operational model can be explained in modern language, using mechanisms of GPCR signaling which we have discovered over the last 40 years or so. Here we have a familiar diagram of the mechanism of cyclic AMP generation by GPCRs. These mechanisms are familiar to most people with undergraduate training in biochemistry or cell biology. In this video, we will explain the operational model with simple language using familiar biochemical concepts. There will be no mathematics here. We can explain how GPCR pharmacology works using some pretty basic assumptions. The goal here is to provide you with the information necessary to explain GPCR signaling pharmacology to other scientists. In the follow-up video you can find on YouTube, we take a deeper dive into the operational model, seeing how powerful it can be to explain our data. Why should we care about this? It helps us solve problems in drug discovery. For example, the operational model explains a result we often see in lead optimization in drug discovery, shown in the graph here. Usually, we test agonists on cells that overexpress the receptor because these assays are straightforward and high throughput. Using cells that overexpress the receptor, we can identify potent full agonists for stimulating signaling. We are excited by the high activity because this means the agonists are potential drug candidates. For example, in green we have compound 1 stimulating cyclic AMP generation in CHO cells overexpressing the GPCR. In this assay, compound 1 is a full agonist with an EC50 here of 1 nanomolar. We also test compounds on model cell lines. Examples of model cell lines include primary cells harvested from tissues and immortalized cell lines derived from these primary cultures. These cells have lower physiological levels of GPCR expression. We typically get to these assays later in the drug discovery process because they can be tricky to develop and are of lower throughput. Very often in these model cells we see the compounds are dramatically less active at these physiological levels of receptor expression compared with our overexpressing cell lines. For example, in model cells, the data in orange here, compound 1 produces a lower maximal cyclic AMP response, and it has an EC50 here of 150 nanomolar, which is 2 orders of magnitude higher than in our high-expressing CHO cells. All of a sudden, our compound doesn't look so appealing anymore. This result can be alarming to people on our project teams who are not familiar with GPCR pharmacology. However, this result can be simply explained if we understand the operational model, and this is why this understanding can be so useful. Imagine being the pharmacologist reporting this result in a project team meeting filled with people who have different skills than we do. Here I'm going to try to give you the information you need to explain this results in simple terms to non-pharmacologists. The operational model provides this explanation. Let's start out here with the basics of GPCR signaling. As you know, when GPCRs are activated by agonists, they stimulate signal transduction cascades. Here we have the cascade for cyclic AMP generation. The GPCR activates the GSG protein, which activates the enzyme adenylyl cyclase, which converts ATP to cyclic AMP. Here we have the concentration response curve for the agonist for generating cyclic AMP. In this familiar experiment, we titrate the agonist compound on the x-axis and plot the resulting cyclic AMP concentration on the y-axis. The concentration response curve is defined by the familiar sigmoid curve when the agonist concentration is on a log scale. Here we have the EC50, the midpoint of the curve, at 10 nanomolar in this case. This is the observed potency of the agonist for stimulating cyclic AMP in these cells. Then here we have the Emax, the maximum signal this agonist can produce in the cells, which in this case is 60 picomoles of cyclic AMP in the assay well. When we first start doing GPCR signaling assays, many of us just assume the data will be like binding assays. We kind of implicitly assume the EC50 of this curve will be defined by the binding of the agonist to the receptor. Let's illustrate what we mean by this. Here we have a population of receptors in blue, some of which have agonists bound to them in red. In this case, 20% of the receptors are occupied by the agonist, so the level of receptor occupancy is 20%. When we first start doing GPCR signaling assays, we implicitly assume the level of receptor occupancy is the only thing that defines the signal we see in our graph here. This in turn leads us to implicitly believe that the EC50 should be the same in all assays. If the EC50 is just defined by the level of occupancy of the receptor by the agonist, then it should be the same no matter what cells or tissues we are using for the GPCR. Then maybe one day we do an experiment with different cell lines. Here is our example from earlier, the same agonist on 4 cell lines with different levels of receptor expression. It can be quite a shock when we first see data like this. We see that the EC50 is no longer constant. Here the EC50 value varies from 1 nanomolar for the high expressing cells to 250 nanomolar for the very low expressing cells. This result demonstrates that in fact the EC50 of the agonist is dependent on the system we use to measure it, specifically the receptor expression level in this example. We need a mechanism that can explain these data. This was the goal of much of pharmacology in the 20th century, trying to understand why the data look the way they do, drawing out plausible mechanisms and testing them against the data. In this diagram here, we have the mechanism of the operational model reproduced from the original paper. Fortunately, the mechanism and assumptions of the operational model are really quite easy to understand, and this is a central point I'm attempting to get across in this video. When we accept these simple assumptions, everything else just falls into place. Many of the assumptions we'll go through here were part of earlier receptor theory, but we will present them all in the context of the operational model to keep things simple. So let's look at the operational model mechanism here. Here we have the agonist-receptor complex. This complex interacts with the signal transduction system. This activation of the signal transduction system then generates a signal. This is how the operational model views the signal transduction process. The agonist-receptor complex couples with the signal transduction machinery to generate the signal. This makes total sense when we compare it with the known mechanisms of GPCR signal transduction. Here is our cyclic AMP pathway diagram from the previous slide. Here is the agonist-receptor complex. Here is the signal transduction system comprised of G protein and adenylyl cyclase. The receptor interacts with this system to generate the signal, which is cyclic AMP in this case. This signal can be anything we like: a second messenger, a muscle contraction, gene transcription, a physiological effect in a clinical trial. The operational model can be applied to all of these agonist effects. The model is so simple. It just assumes the agonist-receptor complex interacts with a signaling system to produce a signaling response. This leads us to the first assumption of the operational model. The agonist-receptor complex acts as an activator of the signal transduction machinery. This assumption is, of course, consistent with what we know about how GPCRs generate signals, as shown in our diagram of cyclic AMP signaling here. This assumption that the agonist-receptor complex is an activator of signaling has a really important consequence. The strength of the signal is dependent on the concentration of the agonist-receptor complex. The more of the agonist-receptor complex we have, the stronger the activation of signaling. This makes sense intuitively, I think. This assumption has in turn another important consequence. The concentration of the complex is dependent on two things: the concentration of the agonist and also the concentration of the receptor. Again, this makes sense. The agonist-receptor complex is comprised of the agonist and the receptor, so the concentration of the complex is dependent on the concentration of both components, the agonist and the receptor. This simple logic leads us to a basic conclusion about how GPCR signaling behaves. The strength of the signal is dependent on the concentration of the receptor. This is what we observe in our experiments. Here is our graph from earlier showing how cyclic AMP signaling increases as we increase the receptor level. This makes total sense in light of our assumptions. If the agonist-receptor complex is an activator of signaling, then the strength of signaling is of course going to be dependent on the concentration of the receptor. It's important to note what we have done here. We have moved beyond the idea that it's just the agonist concentration that defines the response. And now we understand that the receptor concentration matters too. Consequently, this graph is not surprising at all. It is in fact exactly how we expect the system to behave. These basic assumptions can explain something we see in the data here: why the curve shifts when we change the expression level. As we increase the receptor level, the curve shifts to the left. This indicates we need less agonist to generate the signal. Let's simplify this a bit by just using the high and medium level of receptor expression. Now let's look at a specific level of cyclic AMP signal here, at 30 picomoles of cyclic AMP per well. So here's the bit of logic we need to explain this. If we have the same level of signal in the two cell lines, then at this level of signal we must have the same concentration of agonist-receptor complex in the two assays here. This is because we assume the level of signal is dependent on the concentration of agonist-receptor complex in the assay, as we talked about on the previous slide. So, if we have the same level of signal, we must have the same level of agonist-receptor complex. Now, the concentration of agonist-receptor complex is dependent on the concentration of receptor and on the concentration of agonist, as we talked about a moment ago. This means simply that if we have more of one, then we need less of the other. Let's look at this with some numbers. These data were simulated assuming the concentration of receptors in the high-expressing clone was 10 times higher than the level in the medium-expressing clone. The receptor level in the medium-expressing cells is 10,000 receptors per cell, and the level in the high-expressing cells is 100,000 receptors per cell. Now, let's consider the concentration of agonist. For the medium-expressing cells, the agonist concentration we need to get to this level of cyclic AMP here is 10 nanomolar. We can see this illustrated on the graph here. Now, here's the simple explanation. If we have 10 times more receptor in the high-expressing cells, this means we need 10 times less agonist to get the same amount of agonist-receptor complex in the high-expressing cells. This means we need only 1 nanomolar agonist for the high-expressing cells. And here we see this illustrated on the graph. This is simply a bimolecular interaction, so if we have more of one partner, then we need less of the other to get the same concentration of the complex. This explains why we need less agonist in the high-expressing cells to get the same level of signal. And this in turn explains why the curve shifts towards lower agonist concentrations for the high-expressing cells. Now let's look at the next assumption of the operational model. There is a limit to the amount of signal that the signal transduction system can generate. For example, there is a limit to the amount of cyclic AMP that the cells can produce. This makes sense when we look at our diagram of GPCR signal transduction, in this case for cyclic AMP generation. There is only so much cyclic AMP we can generate here. There is only so much G protein, only so much adenylyl cyclase. We could increase the receptor as much as we want, but at some point we reach a ceiling where the receptor concentration is sufficiently high that it saturates the signal generating capacity of the system. There is a physical limit to how much signal that the signal transduction machinery can generate. This again is pretty intuitive, I believe. Let's bring up our graph again. This assumption provides a simple explanation for the results we see. Specifically, it explains why the Emax hits a ceiling as we increase the receptor expression level in our experiment here. We don't just keep increasing the cyclic AMP forever. Eventually, we reach the point here where the receptor is saturating the signal generating capacity of the system. This ceiling is called the system maximum effect, or EM. It is the maximum signal that the signal transduction machinery can produce. Here we have listed out our assumptions again. First, we assume the agonist receptor complex acts as an activator of signal transduction, that it interacts with the signal transduction machinery to generate the signal. From this, it follows that the strength of signaling is dependent on the concentration of agonist receptor complex. This concentration is dependent on the receptor concentration as well as the agonist concentration, and this explains why our signaling results are dependent on the receptor expression level in the cells. Finally, there is a physical limit to the amount of signal that the signal transduction machinery can produce. With enough receptor, we can saturate the signal generating capacity of the system, and this explains why our agonist Emax reaches a ceiling with increasing receptor expression level. So here you see the simple explanation for why the data look the way they do. This is really all we need to understand and explain our result to our colleagues. We don't need mathematics, we just need intuition based on some simple concepts we learned in biochemistry class. Here are some suggestions for drug discovery scientists based on what we have learned here. The first suggestion is awareness. If your team is starting an agonist project, it's a very good thing for everyone to know that the results we get in signaling assays are dependent on the assays we are using, particularly on the expression level of the receptor. The EC50 and Emax are not immutable constants. The second suggestion is to develop these model cell lines assays early on in the drug discovery project. This can take an investment of time, but it has a big payoff in avoiding nasty surprises that can derail our projects. Finally, the use of inducible expression cell lines is recommended. This is now as easy to do as making any other cell line, so there is no cost here. The standard method is the tetracycline inducible expression system. These systems allow the receptor level to be varied, which can be a powerful approach for quantifying agonist pharmacology for translational purposes. Here's a quick preview of part 2 of this presentation, which you can find on YouTube. Here we take a deeper dive into the model to explore how it works. This enables us to understand more deeply why the pharmacology data we obtain from GPCR signaling assays looks the way it does. This video will enable you to use a data simulator which we see here, which is freely available in the link in the comments. This deeper understanding will also provide you with a foundation to engage the literature on the operational model. That brings us to the end of this video. Thank you very much for your interest and attention.